Some of his work, but obviously not all of his contributions, include multicolor single molecule spectroscopy for biomolecular interactions and FRET between more than two molecules. Also some work with SOUR for the introduction of reducing and oxidizing systems for minimizing photobleaching and blinking of fluorescent dyes, and also the description of photoswitchable properties of conventional dyes. In 2009, he won the Academy Prize for Chemistry for the Academy of Wissenschaften zu Göttingen, and in 2010 became a professor of biophysical chemistry at the Technische Universität Braunschweig. Sorry for my German pronunciations. His current research includes single molecule spectroscopy of hybrid nanostructures and multichromorphic systems, single molecule sensing based on DNA nanotechnology, and the development of DNA paint and DNA origami rulers for single molecule and super resolution. Today he'll be talking to us a little bit about single molecule photophysics for super resolution enhanced by DNA nanotechnology. So please join me welcoming Professor Philip Tinnefeld.
Yes, thanks Alia for this introduction, and the longer it gets, the one notices, the older one gets, the longer the introduction. So that's yes. So it's really great to be here. It's a bit unusual format of a meeting and unusual way of putting together, and I found it very interesting. I like very much this format to really think about also the photophysics of the fluorescence markers, and then to see how it develops in chemical biology, and also having maybe now a chemical physics view on that. So I worked a very long time in single molecule photophysics, but recently not so much anymore. So for this lecture I tried to actually for the first time to make it a bit a longer story, and also to put in some new developments of how single molecule photophysics is being viewed, and how we can maybe make fluorescence markers better, thinking about this. So yes, it's an unusual format, and also for me putting really different things together, and I tried to group it a bit into a few things. So talking about single molecule photophysics, and then a bit what we did more recently is our work with DNA nanotech. I don't know whether that is so related to developing fluorescent markers, but maybe that can also be a topic of discussion. So first I would like to it's working. Briefly introduce the group and give a bit an overview, so you understand a bit better what we are thinking. So we don't have this chemical abilities of Kai Johnson. So I was so amazed by this talk how complex sensors he can build. So we are a bit more simple in this sense, but we try to put together like things from different fields and try to create new functions and new ways to look at molecules. Now we are at LMU, so my group is just transferring from Braunschweig to back to Munich, and we are trying to do different things. It's a broad rainbow of things, and one of the things is, so we were involved a bit in this development of the super resolution microscopy, coming from single molecule photophysics, really trying what single molecules are doing, single fluorescent dyes. And one of the things that you needed, of course, after the Betzig paper in 2006, is you needed the abilities to switch single molecules on and off. And so in the old days of single molecule spectroscopy, blinking was always considered bad. So people wanted stable fluorescence, and you want stable FRET signals, and all of a sudden it kind of turned around and the blinking became good. And we were a bit in the middle at this moment, because we were studying blinking quite a bit. And so we could make it useful in a way, and with Markus Sauer then we were involved in this D-Storm development, and also then later on DNA paint and some other ways to switch single molecules on and off, and I will review this a bit, how this is working. So we are not really cell biologists looking at cells. We cannot do that as beautiful as the speakers before. So that is only done in cooperations with people who know what they look at. So we are however a bit of molecular biologists. So Andres, a biology postdoc, is here, joined me, and so we are looking at protein systems mainly in vitro. So we really pick out molecular machines. In this case, it was an RNA polymerase, and it was reconstituted from 12 proteins on a cover slip surface, and then we try to attach with unnatural amino acid fluorescent dyes and do single molecule FRET, and then guide the whole complex through the transcription cycle and see how certain aspects of certain conformational changes occur, and to learn about these biomolecular machines. That was at this time done by Dina Grohmann, who's now professor in Regensburg, and so I'm not the biology specialist, so I will not talk so much about these things. Andres is also much better in these things. He can discuss this. So we really come from single molecules. We always looked at single molecules and want to make it better and ask some fundamental questions about single molecules. Like first we ask a bit more like what is the problem with the fluorescent dyes. Recently we ask more other problems like how can we work with single molecules at higher concentrations, how can we make single molecules much brighter, and ask to overcome some of these fundamental things, and I will briefly discuss this. So that will be like a bit the first part of my talk where I will talk about these photophysics of the dye molecules. And then over the last 7 years we stepped over, we met some people in Munich, and they introduced this DNA origami technique and these origami tools, using DNA as a material and building machines and functional devices. And we got excited by that, and it worked well together if you combine it with single molecules. It changed a bit the paradigm in my group. So in the old days, we tried more here. In this case, you tried to have the most sophisticated single molecule microscopes. So we were working on multicolor thread and having like many APDs and the newest lasers and the newest AOBS and AOTF and whatever. And then alternating and all these fancy things. And with this DNA nanotech now, it turned a bit in my group that now we are thinking more, what do you have to put on your microscope to make things better, and not so much what you build around. And so building nanotechnology tools from DNA to improve single molecule detection. So I will talk a bit about these nano rulers and some other. So we also got into the fields of plasmonics. So now we have to deal with these physicists that do nanophotonics and convince them that also we can do something coming from a completely different direction in this field. And also sensing. We heard beautiful sensor devices this morning. And we think also with DNA nanotech you can create new functions to sense molecular interactions and functionalities. So this is also a direction that is getting a lot stronger in my group now. All right, so brief outline of the talk. So the first part, so this long talk, so the first part I will try to bring you a bit into what really, if you look at single molecules, what single molecules can do, the states and the effects that you see, and looking in some more detail of what was a bit introduced by Luke and yesterday, looking more at the photophysics, and then also the photobleaching problem and what is kind of the status of the photobleaching problem. Going more specifically, it's introduced a reducing and oxidizing system. In the last years, we did not do so much photophysics anymore, but we work quite a bit on the role of thiols, which gives some new ideas how that can develop. And another new development are these self-healing dyes, which I briefly want to discuss, and then also other ways to make blinking and switching molecules, and summarizing that in this context of photophysics. So that will be the second part, and this is of course very strongly motivated by reality. If you have a cell packed with a lot of fluorophores and you take ensemble values, it doesn't matter so much. But if you look at single molecules, or if you do nanotechnology with single molecules, like here was from Hermann Gaub's lab, he developed this beautiful, amazing technique where he takes an atomic force microscope and he can transport a single DNA strand from a depot area into a target area, and he can like draw with single molecules by taking a molecule, putting it somewhere, taking the next one, putting it next to it with nanometer control. An amazing technology. So here we transported these single DNA strands to draw from about 200 DNAs this smiley. It's not so easy. It takes a couple days. You have to do a lot of microfluidics, and it's not so completely easy, but it works. But then, I mean, my job was he cooperated. We were there, so we cooperated. And then my job was to take care that he can also see what he did. So they worked for a few days to build the smiley, and then we look at it and destroy it within seconds. So I think that was not very nice. So there was, of course, a strong motivation to look more into stabilization of these fluorescent molecules. Okay, so then the outline of the second part of the talk is this idea, okay, we built more sophisticated microscopes than this one, but now you can maybe do something with this microscope to make them good by putting something on it. And this is mainly done with the help of DNA nanotech. So how can we improve and also calibrate and better study our, better characterize our new imaging techniques using DNA nanotech. Quantification and traceability is a bit of an issue. How do you really know what you're looking at? I will briefly discuss a bit, but there will be another talk by Jérôme Wenger in a couple of days, how you can more fundamentally change properties of fluorophore. So Luke mentioned yesterday that with these rhodamines, you're sticking to some fundamentals in dye chemistry. And so are there ways to change these fundamentals of like what is the extinction coefficients, what are the quantum yields, and what are the rates, radiative rates, the typical lifetimes? Are there more fundamental ways of changing that? And this, Jérôme will discuss this much more. I will only touch on it. And I will briefly discuss a couple applications of this DNA nanotech devices. One could be like super bright fluorescent beads, very small. And probably I don't come to that. I don't come to the force spectroscopy. That will probably take too long. Okay, so we've seen all the fundamentals of fluorescence microscopy, so I can directly pick you up at looking at single molecules. So of course, if you have an ensemble of molecules, if you have your cells, you see your signal fades away very slowly. But if you look at a single molecule, it will either be there or not be there, so you can do confocal scans. And if you usually get a circular spot, if you have half a spot, the dye is bleached, and the next image it's gone, so you have, it's either there or it's not there. And this you can also see here, it's a transient then over time, you have triplet blinking in this dye, and then at the end it photobleaches, bam, it's gone, and you can take movies, of course, with a camera, and you can have blinking molecules, and then the number of molecules decreases. So that is the classical ways of looking at single molecules. You can do this very easily nowadays. So quite a few years ago, when we started thinking what is this problem of photobleaching, and if you looked into the literature, it was really a magic. There were tons of papers out there. Well, if you add this substance to this dye, it becomes more photostable, but it does not work for that dye, only for this dye. And it was a bit strange. So we tried to think about it more fundamentally, and what you always find in the literature, okay, the triplet state is the evil intermediate. So if you have the dye excited emits, once in a while you have intersystem crossing, it goes to the triplet state, and from there something can happen. And one of the famous thing is that oxygen is involved, so it quenches the triplet by an energy transfer, or like a triplet energy transfer. So you have a reactive singlet oxygen is formed, and that can add to the chromophore, usually forming a peroxide or so, and then your dye is over. And this is of course known for 30 years that this happens, so people started to remove the oxygen. So that was typically done then in the 80s. Also these 1980s, then also enzymatic systems came up to do that in the early 90s, I think. So then the triplets, however, get long. So usually when you have oxygen there are a few microseconds, but when you remove the oxygen, the triplet states goes to milliseconds. And then you have lower brightness for a single molecule. You have a lot of blinking, and the triplets get long, and you can have also other reactions. So you can have excitations from the triplet to higher excited states, and from there have photobleaching. So it's already a state with more energy, so it's more reactive. So it's also not so good. And then people started, I mean, they already added these reducing agents. So they remove oxygen, and the reducing agents was already used before they used oxygen. And then it was all these different chemicals were out there, and it was very strange. So we studied them with single molecules, and you get very ambiguous effects. So like one dye in PBS buffer, it seems quite happy. You have long emission over quite some time, and then you remove the oxygen, or you add a reducer, and it becomes very dark. It bleaches more quickly. It's blinking very strongly. And another dye, you add a reductant. It first blinks, and then you add a bit of a reductant, and it stops blinking and seems to be more happy. So there was no general rule what was happening. So we were thinking, okay, if you remove the oxygen, you are staying in the triplet longer. If you then add a reducing agent, then it should form this radical anion. And if a radical anion it formed, maybe now it depends on the dye. What is the property of this radical anion, whether it will be photobleached more quickly or not, or whether it finds a way to get rid of the electron again and go here. So that was what we thought about could happen. And of course, if you have photo-oxidation, which Luc Levis also mentioned is an important intermediate on photobleaching pathways, then of course the reductant can help. So if that is the most important pathway for photobleaching, the reductant can help. If, however, this is a photobleaching pathway, it would of course not help. So we were wondering what one could do about it. And actually, so reading old literature from Russian spectroscopists from the 70s, which came online, which were not available before. One day I saw they are available, and then I downloaded them all, and they had this ensemble spectroscopic measurements. So they were studying rhodamines and cyanins in a flash photolysis experiment. So you extract with a strong laser, and then you take an absorption spectrum, what you have, and then they say, okay, now I have the triplet state. I add a bit of ascorbic acid. Oh, then I get the radical anion state. Otherwise, I add a bit of methyl viologen as an oxidant, and then I get another spectrum, which is an oxidized state. So all these states, they were shown in the 1970s that they exist for these dye molecules. And then in a side remark, they mentioned, well, then we added both. Well, we didn't get a spectrum. And that was like, that's kind of it. That's the solution. One has to add both, and then all of these states should be saved at the end of the day. So the idea was you need to open all these channels here, that all these states are recovered, and you compete with any photobleaching reaction, and then you can really use the electron transfer to increase photostabilization. So that was the idea. You have to add make all these rates fast, and so add a reducing agent and an oxidizing agent maybe at the same time. So that's why we call that then the reducing and oxidizing system, that you, whenever you're in a triplet, you find a way to go back to S0. So does it work? So with ATO647, for example, one of the brightest dyes in the red, so we remove oxygen always, and always we are working just with dye molecules on DNA. Immobilized, you have a BSA on a glass slide, you have some biotin, neutravidin, DNA, and a dye molecule on it. That's the standard assay. And you remove the oxygen, and you have an oxidant. So then you see this kind of blinking. So this is a trace. This is a zoom in. You see it bleaches quickly. You have a lot of blinking. You can do an autocorrelation to characterize its blinking, and it's about like 30 milliseconds off. So what we believe is happening, it goes to an radical cationic state for when it's in the triplet, and then it might photobleach after here 7 seconds. You alternatively can add a reductant like ascorbic acid, and then you get a bit different blinking, but it also bleaches quickly. It has a bit longer off times. Here what we believe is happening, it goes from the triplet to a radical anionic state, and from there you can also then after some time photobleach, or it finds a way. So it finds a few times, it finds a way to go back, but then after some seconds you have a photobleaching pathway. And if you add both at the same time, the reducing and the oxidizing agent, so in this case it was ascorbic acid with methyl viologen, then the fluorescence gets very stable, and you have the autocorrelation, it's gone. There's down to a microsecond, there's nothing. You see here these two intensity steps, so that is two spectrally different states of ATO647N. This is something that we see for all rhodamin derivatives. They all have spectrally different states. At least when they're photostable enough, then you can see it. So they change like 20 nanometers their spectrum from time to time. And so it works. So does it make sense? So it sounds like a paradox to add a reductant and an oxidant at the same time. So does it at all make sense? So what is actually the rationale behind it if you think more physical-chemically about it? And what you have to look at is, you have to look at the redox potential. And this is a bit what we find is another very important property of a dye molecule. So yesterday we heard you have the brightness, extinction coefficients, quantum yield, the absorption and emission wavelengths, and we think that the redox potential is also another very important parameter to characterize the dyes. So we characterize some of the dyes. In the meantime, quite a few dyes have been characterized, and they're like quite general rules that you know. Looking at a dye, you roughly know in what range these redox potentials are, and so then you can look at these reactions which are occurring. So what are you doing? You're giving the system in this point an energy of 1.88 electron volt, which is a 0-0 transition, the energy of the photon that the dye then has. Then also from the literature you find the energy of the triplet state of Psi5, for example. And then we also measured the redox potentials of the Psi5. So it means kind of you have to apply 0.97 electron volt, and then Psi5 will start oxidizing, sorry, other way around, reducing. No, that's should be there, right? So reducting and the oxidation potential. To oxidize it or to reduce it. So you find all these potentials, or you can measure them, or meanwhile you found most of them in the literature. And so you know all these energies that you have to give the dye to get rid, to give it an electron or to take it away. And there's this formula which is called the Rehm-Vela equation, where you combine all these energies and calculate the delta G of essentially each electron transfer reaction that can occur. So for each step, then, if you know all these energies, you can calculate how your delta G is for a triplet reduced by AA. So you also need the potential, the oxidation potential of ascorbic acid to give away an electron, and the of methyl viologen, the reduction potential to take up an electron. You just plug them all into this equation. So the energy you give by the photon, because it's a photo-induced energy transfer, the reducing potential, for example, of the dye, the oxidizing potential of the reducer. And the C is a constant from Rehm-Vela equation, and that is, well, the product of the reaction is a radical pair. So you have a dye molecule and a radical next to it, and they have an interaction. And this interaction usually stabilizes the radical pair, but also it plays a strong, it's very important what the dielectric constant is that shields this interaction between the radical pair. So this is close to 0 in water, because water has such a high dielectric constant. So you can even neglect this, and then you have a very simple equation, and you see all these terms are negative. So all these reactions happen for Psi5, and you can also go through this state or through this state. Thermodynamically, it's all easily accessible and easily calculatable. And so how can you see that? At the end of the day, this reaction, the dye is a catalyst. It's a photocatalyst. So the dye, the energy comes from the photon, and then it catalyzes the reaction of transferring an electron from ascorbic acid to methyl viologen. So usually in solution, they are so mild oxidant and reductant that they would not react. But because of this light energy that you put there, the dye takes the electron from the one and gives it to the other. So it's, from chemical point of view, it's a photocatalysis occurring of the dye molecule. So photocatalysis is also a very popular topic in chemistry nowadays, and it took us a while to realize that this is actually a photocatalysis. Okay, so that helped a lot to understand a bit better the dye molecules. So now we learned how to have them blinking. For example, in this case, it's not a triplet state, it's a radical state. And you can make them well, you can stop the blinking by just adding the substance which would save this radical state. And if you remove oxygen and add everything for At647N in this case, you can get millions of photon on average for each single molecule. That works nice. It does work for all dyes, and of course there are exceptions to the rules. And so one dye that puzzled us quite sometimes is this. It's At655. It's an oxyzyme. And these oxyzymes are known to have a high electron affinity. So also when you take this dye and attach it to a DNA, you can have photoinduced electron transfer from guanosine to this dye molecule. So it really has a low-lying radical anionic state. And actually it has a high-lying oxidized state. So you can only go this way. You cannot go through the oxidized state. This is also one of the reasons why this dye is incredibly photostable in the presence of oxygen, because, as Luke mentioned, the radical cationic state is an important intermediate in photobleaching. And for this one, it's almost inaccessible. It is very high in energy. And this is why this dye is probably one of the most photostable dyes in the presence of oxygen. But it has a very lying radical anionic state, so when we add our typical substances, you still see blinking. We could not remove the blinking for this dye molecule with the Redox system, and the reason is, you can calculate that you have a positive delta G for this reaction. So the energy from the radical, this energy is not enough to drive this reaction. It's slightly endergonic, and this means it doesn't mean it doesn't happen. It just means it goes slower, and you still have blinking under all these conditions. And so for ATO655, this means you have such a stable radical anionic state that you can really switch it, and it can be switched off for quite some time. And this we showed here. So we have a green dye, so we know where the ATO647 is. So the ATO647 is in the 655 is in red, and you have these yellow spots, single molecules, and then you remove oxygen and add a bit of ascorbic acid, and all the red dyes are gone. The green one is now blinking because you have the radical anion. And then you add a bit of oxygen, you let a bit of oxygen come in, and all these dye molecules come back. And then you can switch it off and on and off and on. And many, many times it's very photostable. 1,000 cycles you can easily do. And you can very nicely control it, the on and the off states. So you can control with methyl viologen, you can control the duration of the off state, and the on state stays constant. And with ascorbic acid, you can control the duration of the on state, and the off state stays constant. So you have independent handles to tune that, which you can use for super resolution microscopy. And it also works in the presence of oxygen. And this is also the reason why Markus Sauer's group then used this dye for the first live cell localization microscopy experiments with organic dye molecules, because this works under essentially all conditions, and this is still for live cell blinking, when you need blinking molecules working. I don't know, probably still best. I don't know how other versions there are, if you just use the blinking. Okay, so you can now use different offstates if you want to have blinking for super resolution microscopy. So for Psi5, it's more known, the D-storm, that you have a chemical reaction, and I come to that when I talk about thiols. But you also have other dark states of Psi5. So you have the triplet states. It's about 15 milliseconds, and it was claimed by some people you can use triplet states for localization microscopy, and that is not really true, because 15 milliseconds is not off long enough that for a densely labeled structure you could localize many molecules in that time. So 15 milliseconds should be too short. But so in most cases you need something longer lived. For Psi5, the longest radical anionic state you can get by increasing ascorbic acid concentration is about 60 milliseconds. And with that you can do a bit of, well, you have to do quick super resolution, so that 60 milliseconds off state is enough. But it is possible, so we showed that you can do it. And so you can have all molecules blinking, and of course this was then used for the super resolution microscopy of localizing molecules. And this is actually an example where we had Psi5 radical blinking. So it's not D-storm, because we use the D-storm use the thiol-induced switching, and this is really the radical anion, and it's 1 millisecond per frame integration time, so very fast. And in 1 or 2 seconds you go through this, you localize all the molecules, and then after a few seconds you have an image like that. That's when you use the radical state and not this long time. So that's why we called this blink microscopy back then. All right, so yes, so these are old images of just cytoskeleton proving that this works. And so one of the open questions was actually in the 1980s, I stepped over a paper by John Sedat, where he just showed that you have better photostabilization with N-propyl galate than other chemicals, and it was a science paper in 1982. And also in 2006, Tekshipal just showed that Trolox was better than many substances for single molecule measurements, and you get more stable emission. So we also tried to publish this in Nature Methods, the ROX method, but it didn't work. And the reason why it did not work was mainly because we did not get much better than Trolox. So the ROX was like, we knew what we were doing, so at the end we had like some kind of understanding, but from a practical perspective, we were like equally good as they were. So they had just a single substance, Trolox, showed you get very stable molecules, and we got pretty much the same results with our ROX system. So we were puzzled what was going on. And then later we looked again at it, and we had like many theories what could happen. Why could a single substance be as good? Because Trolox is also reductant. It should do photo-induced electron transfer. So what happens to the radical anion then? So we looked at it and had many theories what we could do. So one thing we noticed was the following. So we used the ATO655 again now as a redox sensor to feel the environment. So it blinks, the ATO655. It goes in the off states because Trolox is a reductant, and so you have the radical anionic state, and then the dye is emitting. And what we noticed, if we measure the same thing a few hours later, the blinking would change. The off states get shorter and shorter. So it goes like this. It gets shorter and shorter, and it's almost gone after some time. So this is in the beginning, freshly prepared Trolox, and then you measure after some hours, and then it looks like that. So what we found out is the on counts are constant, but the off times are decreasing over time, over about 18 hours. And so then we didn't know what was happening. We were digging in the literature, what happens to Trolox when it degrades maybe, and we found in Journal of Food Science, we found an article which was telling us that Trolox was oxidizing over time to Trolox quinone. And this is very important. Trolox is used as to describe a reductive capacity. You have Trolox as a reference, and so there's a lot of study about Trolox. And so with UV light and also over time with oxygen, you get this quinone. And the quinone is, of course, a classic oxidant. And here you have the reductant. And you can follow this reaction very easily. So here you see how when you oxidize it, how this band disappears and a new band comes up at 260 nanometers. And just from taking the spectrum, you can directly see how much Trolox you have and how much Trolox quinone you have. So that was solving it. So at the end of the day, Trolox is also works according to rocks, and you can also understand why it gets so photostable with rocks. Okay, so that is like now the classic photophysics, how we now kind of understand how these dye molecules work and under which conditions you can make them photostable. There's a bit of special role of some reductants, and one of these reductants are typical thiols, like thiolethanol or mercaptoethylamine. And so one of these things that we discovered, and to be honest, it was Emmanuel Majat, one of our hosts here, who first discovered it. So when we were both working in Shimon Weiss' lab as postdocs, so one of the things we noticed when you look at Psi5, and we wanted to do that for Fred experiment, and we had added some substances to make it more stable and removed the oxygen with the glucose oxidase system, then you sometimes it goes off. But then if you switch on the green laser to do the Fred experiment, well, first, no, that was not, it was bleaching, it was just quickly bleaching. But then you switched on the green laser, and you switched on again the red laser, the dye was back. So that was very puzzling at that time, and the people in Shimon Weiss' lab, they were not so interested in the photophysics, but in the RNA polymerase, what that was doing in the Fred experiment. So a couple of years later, then we picked up the idea and said we have to look at that more closely and characterize it. And so that was the Psi5 that can be switched on and off that we discovered by that. And so it was not clear what was going on there. And a few years later, the Zhuang lab, so they had published also in the same year, 2005, this paper where you can switch Psi5, but only in the presence of an activator, the Psi3 Psi5 couple. That was a couple of months after this paper published. And they came up then 2009 with this JAX paper having a mechanism that you have a nucleophilic addition of a thiol to the polymethene chain of the Psi5. And this, well, so our approach without the activator then led from Markus Sauer, the D-Storm superresolution technique. So thiols are different, so they can cause these long off states, and you can use it for switching, and also some other mild reductants like TSAP does similar thing as shown in another paper by the Zhuang lab in 2011. So you can have different mechanisms of thiols than ROX has, so that's certainly something different. A question is, how another question came up then. So thiols then were a bit mystical for us. So all the dyes look different in the presence of thiols, and you have the long off states, but there was something else. And this I want to describe you now what higher level of photophysics you can find. So just summarizing rocks is you have to get rid of the triplet. You have these molecules where in the presence of oxygen they bleach quite quickly, Psi5 and PBS. You can be happy if you have a second. If you remove the oxygen, you get the triplet blinking. So that's really the triplet blinking, 15 milliseconds off state of the Psi5. Then you add the rock system. Psi5 gets very happy. It lives for up to a minute or at considerable count rate. So the dye is very happy. But to be honest, it does not work for all the dyes, right? So I don't know if you work with green or blue dyes, it doesn't work so well. So what's going on there? First question, and second, what can you do about it? One of the things is rocks is always diffusion controlled. So it can never be faster than how fast a dye will collide with these reducing and oxidizing molecules. So it cannot be fast. If something happens faster than these collisions occur, you cannot get a handle on the dye. So what could happen is, for example, you could have an excitation to the singlet state, and from the singlet state you can have an higher excitation to a higher singlet state, or something like this, which would be faster than you can have the collisions. And if that is an important bleaching mechanism, rocks cannot help it. And it happens apparently more the shorter you go with your excitation wavelengths. You have more energy in your photons, so it seems to become more likely that you have other side reactions. So we are a bit limited to shorter wavelengths with everything that is diffusion controlled. And Alexa 568 is an example that did not work so well with rocks. And kind of accidentally we saw that if you have, however, I come to this, to the solution. So that, well, at the end we found that with thiols it gets much better. So we did not know what was going on. So the question is, what is thiols versus the reducing agents? I mean, thiols at the end of the day are also a reducing agent, especially in the thiolate form, in the deprotonated form. So we characterize it again by the blinking. So we were looking for a mechanism, a photochemical mechanism, just from looking at blinking molecules. So we characterized this dye in the presence of like a reductant. We increased the concentration of ascorbic acid. And you see that the blinking, that the on times get shorter. So when you go through the triplet, that you don't see, it's relatively short, but if you go to radical anion, this is this blinking that you see. And these reactions compete, and if you add more ascorbic acid, it rather goes this way than this way. And this is why these on states get shorter. You can see it by eye easily. So there are very short spikes. You have long on states. That's more ascorbic acid, more radical anion states. If you do the same thing with mecaptoethanol, it looks very different. And mecaptoethanol is, we could show it's the active form is like the deprotonated form S minus, and because from the pH dependence, and so it lives longer, it blinks a bit, but the appearance of the blinking stays the same. It remains exactly the same. So we were really puzzled what could be going on. So we had to look more closer. One thing is you do autocorrelation. And in the autocorrelation you usually see a B exponential decay. So two processes happening. And one process is influenced by the BME. The other one is not. So this is a radical state. And this one gets smaller and smaller the more BME you add. So the triplet is depopulated. But this state, as we assigned to the radical anion, is not populated more. It's the ratios is staying the same. So the off states are the same number. So must be another triplet depopulation pathway. So it was puzzling us quite some time until we had an idea. And so to make a long story short, so we had to do many experiments to get to it. The solution is that we have what is called a Germination Recombination mechanism. So what does usually happen? Usually when your reductant comes, you go to a radical anionic state. The dye molecule goes here. It's the ascorbic acid, or what it was oxidized and swims away. You have a radical anionic state that has a long lifetime if nothing oxidizes it. Now what is thiol doing? The thiol is doing something else. It is also reducing the dye, but it is not always swimming away. It is only swimming away in 1% of the cases. And in 99% of the cases, it's staying there and giving the electron back, taking the electron back. Sorry, taking the electron back from the dye. So first it gives the electron to the dye, but before swimming away, it takes it back in the ping pong mechanism. So 99% of the time it does this, what is called geminate recombination, and only 1% of the time you get the radical anion. This is the reason why C is infrequent blinking. And since this is a ratio which is independent of the concentration of the thiol, you don't see a change of the appearance of the fluorescent trace. The time trace always looks the same because the ratio stays the same. The probability to go here and here stays the same. So that is, at the end of the day, the solution of what is good. And this is, of course, pointing also towards new ways of increasing the photostability. If you have something that remains there and does this ping pong mechanism, that would, of course, help. And you can see it directly in these transients. So this is, yes, now we combined only rocks with these green dyes, and you can see it bleaches. And if you have mercaptoethanol combination with rocks and oxygen removal, dye molecules stay much longer and also almost don't blink. You have still just a little bit of blinking, but it helps quite a bit. Okay, so that was how complicated it can get, and what you can get from these single molecule transients. So another question is, of course, why are thiols different? So why does ascorbic acid give the electron and then swim away and thiols don't? And there was a paper recently from the COSA lab last year in which they were discussing the reason why thiols might be different. So what you have is you first have your dye in your singlet state. It goes to the triplet state, and then you have the electron transfer reaction to the triplet state. But what happens is that these electron spins, they stay correlated. So you still have like a triplet radical ion pair. So they cannot recombine because they are still correlated. They could only go back to the triplet state. So you have a correlation of these spins. What you need is you need intersystem crossing. So one of the two has to do intersystem crossing, and then they can recombine. So you need an intersystem crossing by spin-orbit coupling or whatever to then have the recombination. And it is known from the literature that thiols, especially when the charge is, or the spin is really localized on the thiol itself, they have a much, much higher intersystem crossing rate than other molecules where it's more delocalized, like especially ascorbic acid or something like this. So the reason is that the thiol does this intersystem crossing faster, so it can do this geminate recombination in a single step. Okay, so that directly brings me to how could you now make dye molecules better? And there was a paper from Scott Blanchard's group in 2011 where he said, well, if you can have like this rock system, so apparently had heard about that, so if you can add a triplet quencher directly to a dye, then you could be more efficient, and especially it could also work in cells where you cannot control the redox medium. So he synthesizes Cy5, so you can couple it to a biomolecule, and via a linker he has a triplet state quencher, and he also uses Trolox, for example, to bind to it. He uses an electron acceptor, and also COT, which does a triplet energy transfer to the dye molecules. And essentially in all cases he gets an improvement of the photostability. So he wanted to make an internal self-healing, well, he didn't call it self-healing at that time, but he wanted to make an internal protector for the dye. And it kind of worked, but interestingly he never got as good as a solution rock system. So he got better, but never as good as what you could do when you had the solution. And one of the reasons, what we believe could be the reason, is that you need this intersystem crossing. So it's the idea, that is what the mechanism that was not proposed in that paper, that the mechanism how a single molecule could heal. Torben Cordes and I suggested that you have like an internal rock system in which you have a geminate recombination mechanism. So instead of having two molecules in the solution rocks where you do reduction and then oxidation, for example, now it's the same Trolox molecule, for example, that does the electron transfer first, and then they have a back electron transfer. So here it is forced to do a geminate recombination mechanism. But now after 5 or 6 years, so the self-healing dyes could not become better than the normal dyes so far, and we believe that the main reason is probably that you need an intersystem crossing, that you can have a quick recombination. Otherwise, the radical ion pair will there for quite some time until one of the two does this this is recombination. And this is, I think, a step where improvements could happen, that you have to put something there that does this intersystem crossing. So my suggestion to you. All right. Okay, so from this I want to switch gears a bit. I have a bit of time still. So and talk about more this DNA nanotech applications that we've recently been developing. So it really started also from looking at super resolution images and going to the conferences around 2008-9. And this was when there was the big hype starting about Palm, Storm, and also of course STAD. And so you went to the conferences, so we also showed our ways of switching molecules, but everybody of course showed filaments, so either tubulins, actins, whatever. So everybody had new ways of labeling, new ways of doing whatever, but the images you saw at the end of the day, they were always filaments. So it was very hard to say, whew, how good does it really work? So how many dyes are there? How many bleaching is there? How was the labeling done? How big are these antibodies increasing the size of the structure? What is really the resolution? So you zoom in wherever you want, and maybe it was noisy on your side and you get, I don't know, 80 nanometers, 90 nanometers resolution. I don't know. It was just not defined. And so that was, so you sat there and you never knew what system, how good is the method really, how good does it really work? I mean, you sometimes find a good thing in the cell. It's hard to make cells reproducible, that they all look good with the microtubules and everything. So there was a moment when we ran into Fritz Simmel's group from Technical University of Munich. And he at this time, he had established in his group this DNA origami technique. So they were like, well, we can now do this DNA origami, but what do we do with it? And we were there like, well, we are now doing the super resolution, but well, how good is it really? So we were thinking, can you use DNA nanotech to place fluorophores with nanometer accuracy? And so this DNA nanotech field, it is from the 1980s. It started with Ned Seaman, and I don't want to go into the history, but the key paper for us was this Paul Rothermund paper, which made it really practical. So before that, people really had many authors needed to make one paper. Now it was one author. It was a single author Nature paper of cover. And so you needed many authors first to make one DNA structure, and now all of a sudden there was a single author who made many DNA structures. So something had changed in this moment, and so they're big. They're like 80 times 80 nanometers in size, made from DNA, and you get overview images, and you see the yields are really outstanding and many structures. So it was really a game changer in this field. And so how does it work? It's quite simple. You have a long strand that is taken from a phage, so you can isolate it from a phage. You know the sequence. And you start adding short strands, so you know the sequence, so you can design to make a loop here just by adding a DNA strand which hybridizes here and there. And then you add not only one of these short strands, but you add more, 2, 3, 200. And you get to a predefined structure. So it's only programmed by the sequences of these short stapled strands, they're called, that this comes out as a rectangle or square or whatever. And of course, there's all the DNA chemistry out there that you can buy dye-labeled DNA or however chemical modified DNA. So you can easily place dye molecules wherever you want on this breadboard. So we never invented anything of DNA nanotechnology. We only used it. So for us, it's at the end of the day, it's a molecular breadboard. So you can pin whatever you want at whatever position to your DNA origami. And forget even about the DNA structure underlying it. And it's very simple to do. You buy or you make the scaffold, you add the 200 stapled strands, you heat, you cool, you run a gel. You can add dyes, proteins, gold particles. You have atomic force microscopy and electron microscopy to characterize it. So you get tons of structures which are all identical, super big yields. You can see individual strands missing in atomic force microscopy. So it's really a very nice tool meanwhile. And so we are now developing a lot, trying to build things from this. So that is a bit the new direction in my group. And the first thing is actually then the nano rulers, first with Fritz and then also with Tim Lille, and so that you can build nano rulers. Oh, there's one transparency missing where I showed the stat nano rulers. So we started actually with single molecule localizations. So you can place two molecules at a distance of 90 nanometers. So instead of getting a blob, you get two spots at a distance of exactly 90 nanometers. That's how we started. And then of course we were thinking, well, what source of microscopies it could be useful for. You can make it for a confocal microscope. You have a distance of about 300. Well, that's the contour distance. So the real distance is about 360 or 50 nanometers. And you can see it in the normal microscope. This dumbbell structures. With that microscopy, you can go here. We have 71 nanometers in this case, but meanwhile it was shown below 20 nanometers even with DNA nanostructures. So this is the confocal image, and you can tune, you can increase the power of the stat beam, make these spots smaller until you see the double spots. So that's a nice thing about the stat, that you can tune the resolution really. And so you can also do fluorescence resonance energy transfer, and so place a donor and an acceptor dye at different distances and measure the R⁶ dependence use it as a ruler. So you cannot do it so easily with double-stranded DNA because of the helicity of the DNA. You always have also the distance changes because you're on the other side of the DNA. But with this, if you always put them on the back of the helices, you can really precisely measure this R⁶ dependence on the back of a DNA ruler. And you can make 3D structures. So you have a dye down here, you make a pillar, it's with biotins bound to the surface, you have a dye up there. You use, for example, this astigmatism introduced by Xiaowei Zhuang's lab, and you can localize the dye up here and down here, and you get distributions of how molecules are oriented. Alright, and also you can interact other interactions, a plasmonic ruler which has a longer distance range, distance dependence to nanoparticles, and so forth and so on. You can go down to 6 nanometers in this case, so you can measure distances on the origami 6 nanometers. Meanwhile, Ralf Jungmann showed even smaller distances, 5 nanometers, and with better localization precision. And this is done by DNA paint. So coming back to our fluorescent markers, to our fluorescent probes, if you use blinking molecules, or you of course you split your photon budget in many on states. So maybe photoactivatable molecules are better, but then you cannot see your structure before. So they're dark before. So it would of course be great if you get rid of this photobleaching problem and really get every marked position with a molecule. And one way to do that would be if you could replace your molecule on your dye molecule on your label. So one thing you could do is, for example, you could think of labeling your structure with a short DNA strand. And this DNA strands you could then make so short that the hybridization is not stable. So instead of using 20 nucleotides, maybe only using 8, 9 nucleotides, and then when a labeled strand comes, it will bind to it, and then you get a bright spot, and then it's not thermally stable. So after 1 second or so, it will diffuse away, and the next molecule can come and can replace this dye molecule. So what you would do is you would label, instead of with a dye or with a tag, you would label with a short DNA strand, which provides a designed interaction strength for how long this molecule should be there. And not only can you replace the dye molecules to reduce the bleaching, but you could also use this really directly as blinking. So you can make the concentration of these background molecules so low that that only once in a while a molecule binds, and you don't get so much background, especially in TF, or if you use a bit of mechanism which makes these molecules in solutions darker, and then you only get a bright spot when they bind, and this creates you a blinking like this. So it unbinds, it binds, and unbinds, and it binds. And the advantage is, of course, that you can use a full photon budget. You can take the full number of photons you get from this dye molecules when it is bound. So for some dyes, it can be more than 30,000-40,000 photons on average. And you have no photobleaching problem. You can control the kinetics by the concentration in the solution and also by the length of the DNA strand. And you can use the full photon budget of the best dyes. And the best dyes is not completely true. The dye should, of course, not be sticky. For example, Atto647N is not working so well, except if you have very good surfaces which do not stick, because it's too sticky. So that's a bit of the problem. But with that, you can see this how with the length of the DNA, for example, the time until it binds stays constant, but with the concentration until it binds, it is going smaller, but the time it is bound is completely constant. So you can tune these rates by concentration and the length of the DNAs with this DNA paint. So Ralf Jungmann, then postdoc at Pangying and now independent group in the Max Planck, also professor at LMU, has developed this much further in the last couple of years. So we've also used it a lot now, and so we built, for example, this ruler, which has a distance of binding sites for the dye molecules of a distance of 80 nanometers in this case. And it works so robustly, so you can have thousands and thousands of structures on a surface, and you get such an image in 5 minutes. So it's super easy. All of them have like 3 dots, 80 nanometer distances, and this was a motivation for some people of my group to found a company, Getaquant. They are now commercializing that. So I think these are really good training structures. Especially you don't always want to calibrate your microscope, that you can do your pixel size and everything. But if you're not so experienced with super resolution microscopy and blinking single molecules, this is, I guess, the easiest way to do it. You only have to take the samples, put it on the microscope, and get this blinking from this binding and unbinding of molecules. So that works really nicely. So a more recent development that we just uploaded on bioRxiv is nanorulers for expansion microscopy. So expansion microscopy is really also very exciting microscopy from Ed Boyden's lab. And the idea is, you probably have heard about it, that you don't use some crazy optics to get your magnification better, but instead what you do is you label your sample, then you remove your sample, but you leave the labels in there by putting a gel, and then you swell the gel and just magnify the sample. And from this you can get from swelling you have a magnification. So this also, we wondered whether that works with DNA origamis, and whether that can work quantitatively. So how would that look? And so we made a DNA origami and labeled it with this acridite. So we had a counter sequence with an acridite on the one hand side and a dye molecule on the other side, and then bound it to this DNA origami platform on the surface. So you have DNA origamis on the surface. They have a dye molecule and an acridite chemistry on the strand. In a normal TIFF image, these two origami here would just give you big spots. The distance is about 120 nanometers. But then, if you make this gelation, you make this polymer gel in there, and during this reaction, you connect the acridite to the polymer, and so it connects this strand with a dye molecule to the polymer. In the next step, you denature, you remove all the DNA from the sample. All the biomolecules are gone, but the dye molecules are still attached to the gel backbone, and then you expand it by changing the salt concentration, and then you image it, and you should get these 2 spots instead of the 1 spot. That was the idea, and we wondered whether that could really work so robustly that you can do that with DNA origamis. And so this is how it looks. So this is a gel in the beginning before swelling at 5.4 millimeters, and then you swell it and it becomes really big. And you have to play a bit that it stays on the surface, and it is almost 20 millimeters. So you have a macroscopic expansion factor of 3.6 with this technique. And then if you look in the TIFF image first, in this case you get all these single spots, and then you do the expansion and you really get double spots. And if you look at the intensities we cannot completely quantify, but it seems like they are really almost, if you take the 2 spots together, they are almost as bright as them. So it almost seems to be almost quantitative, this binding of this acridite to the gel in the background. And from the magnification, now you can resolve these structures before you could not resolve them. And from that we could also measure the size of the structures before and after the expansion. And we achieve a macroscopic, so now we can measure locally and say, well, how is really the magnification at the surface? And we find that it's substantially smaller than the macroscopic expansion factor. And that's, of course, one of the questions that people who do expansion microscopy often get, like, well, how is really is this macroscopic expansion that you see, is it also reflected on this molecular scale? And is it also isotropic? Is it isotropic in all dimensions? And we cannot answer many of these questions yet, so we're just starting in this direction. But here we see this discrepancy, and I think this discrepancy is not surprising, because these origamis were immobilized on the surface. So if you imagine how it swells, you have the shear at the surface, so it appears quite likely that at the surface it is not expanding the same as in the volume. But on the other hand, I think it's important to be able to characterize that, and this is the first step of being able to characterize really what expansion microscopy is doing. And in principle, the data show that the technique fundamentally really works great, so I think it's a great approach, but we have to do some more characterization of isotropy, for example. Okay, so another application which is also very simple, what you can do with DNA origami is you can think of making brighter point sources and even brighter labels by putting many dye molecules on an origami. And this becomes more important nowadays with super resolution approaches, because people for many years didn't care so much. They were taking some fluorescent beads to characterize the microscope, and you put a fluorescent bead. It didn't matter so much if it was 50 or 100 nanometers, because your PSF was almost much bigger. But with stat microscopy, when your PSF gets very small, then the size of the bead matters. How big is your bead loaded with fluorescent dye molecules? And if you look at also how these beads, so it would be good to shrink it, to have smaller beads, but of course they should not be much darker. And if you also look at the small beads that you can commercially buy, like 40 nanometer beads, they are very inhomogeneous. So some of them are very dark, some of them are very bright, some of them seem aggregated. So these beads, 20, 25 nanometers, or up to 40 nanometers, they're everything but perfect. If you go higher, they get more homogeneous and better, but below 100 nanometers, these plastic beads are not so good. So the question is, can one do better by putting dye molecules in origami and placing them densely? And the question is, of course, what are the reasons why they are so inhomogeneous, these polymer beads? And one of the reasons is, of course, when two dye molecules come too close and they start interacting with each other, they will start forming aggregates. They will not be the single dye molecule, but they will be an aggregate of dye molecules, and those are usually not emitting. So the question is, how densely can you put dye molecules before they start interacting, for example, in an origami? And so what we started doing, we placed two dye molecules at different distances in this origami, one base pair, put it on the origami, have some reference dye to find the structure, that we know where the origamis are, and then change the distance, like 2, 3, 4, 5, 6, have a larger distance, 11 base pairs on the origami, and then see what happens. So to see how bright can one fundamentally make such a structure. And this is how it looks on the single molecule level. So in green is where we know where the origamis are, and then we have one base pair. They are dark. They are absolutely gone. 2, you see a little bit of blinking. 3, some shining up. At 4, they come up. They blink. 5, 6, they are bright. 7, they are bright. And this is the colocalization then for the 7. So then they are bright. So you need a certain distance that these dye molecules don't interact with each other. And so if you take now, well, there's very interesting photophysics in there. For example, the fluorescence lifetime is changing at a different distance than the intensity is. So there's quite something going on photophysically, but I don't want to go into these details. But fundamentally speaking, what is the density with current linker lengths and dye molecules? How many molecules can you place? And we can calculate that easily 500 dye molecules should be possible to put in a container of 30 times 30 times 30 nanometers. And this would be like about 100 or not, 20 to 50 times brighter than what you currently get for this size of fluorescent beads. So this kind of should work, and also Germination one has tried to go a bit in this direction. At the moment they are at 80 dye molecules, but that was already enough to beat these 40 nanometer beads. They are much more homogeneous. They are on average brighter. They have homogeneous fluorescence lifetime, all kinds of properties. And together with SVI and Leica, they could show that deconvolution of stat point spread functions works much better with these beads when you have an experimental point spread function. So you can resolve these, that these are 2 beats, only if you feed the deconvolution algorithm with with the correct PSF. So that is SVI who are producing this deconvolution software, and only with these beats they could get the right structure. All right, so it brings me to the almost the last application, which I only briefly want to touch, and I only want to discuss a couple more fundamental ideas of our limitations developing fluorescent dyes. And this is fluorescence amplification using DNA nanotech and antennas. And as I mentioned, I briefly want to touch it and have a few thoughts on it, and we hear about it more on Thursday. So the fundamental idea is, when we think of fluorescent dyes, they are limited in the cross-section due to how the physical chemistry and the quantum mechanics is. You would have them much longer to get better cross-sections and things like that. And some people say that nanophotonics is a field that is developing much more dynamic in many directions, since you can make nanoparticles in very different shapes nowadays with very different materials. So maybe that would be a way to do, but you can also couple it and combine it with fluorescence. And the fundamental, very simple idea is that the E field of your excitation beam, it can induce a polarization in your particle, and you get an oscillation, and this is called this resonance oscillation is called a localized surface plasmon, and this kind of stores the energy in your oscillation, and this makes a higher E field here and here. And if you have a dimer of particles, you get a much higher E field, and you can now have a small near field volume, which is only 20 times 20 nanometers in size, where you have a very strong excitation intensity. And to realize this, we used DNA origami to place the dye molecule there, and then to place these nanoparticles. And so this is how it looks in the EM with the DNA origami particles, and they have a gap of 20 nanometers, and you don't need lithography and anything. So this is the old dye blinking zoomed out. So this is the old dye blinking zoomed out. You can place a dye molecule here in the middle, and then you place one particle, the intensity goes up by about 8-fold. And then you place 2 nanoparticles, and the intensity goes up by another 10-fold or so. So you can make single dye molecules extremely bright. And this in our group now goes into many different directions. And I want to only mention a couple things, which is, well, can that be a label? Can you make a new label with that, which you would ask as when you think about fluorescence markers for microscopy? And the answer is, well, probably not really, because these particles are like 80 or 100 nanometer in size. So I think a label with 80 or 100 nanometer in size is not so convincing. On the other hand, well, I think you could make like sensors. You could really maybe place something like this inside a cell and have a sensor in the hotspot, and it would report you super sensitively with just minimal light excitation on it. So I believe for sensing applications, this is very promising. So another thing is, if you think about photophysical rates, this is always when you have a fluorescence marker next to metal nanostructures, you can change all the rates, everything, all the properties of the dye molecules change. And like the excitation changes because you have the higher electric field, but not only that, so also the radiative rate changes and the non-radiative rate. All these rates change substantially. The intersystem crossings should in first approximation not change, but in second maybe it does. And we wondered whether one can characterize this, because of course if you can manipulate these rates, this can give you new opportunities to design fluorophores and think about how you can change that. And I don't want to go into detail, just mentioning that we found a way to do that using the blinking, because 3 rates are changing and you need 3 parameters to measure. So we use the fluorescence intensity, the fluorescence lifetime, and now also the number of photons as third parameter to be able to extract all the 3 rates. And then you can go through your fluorescent transients, and you can determine quantum yields of single molecule next to nanostructures if you follow this recipe. I don't want to do that, but we did this next to nanoparticles, and we did simulations, electrodynamic simulations, and we could fairly well recapitulate all these properties. You can really get all photophysical rates by analyzing single molecule blinking next to the nanostructure at the end of the day. So another direction that we are going is really sensing applications. So at one day, we don't if we can make dyes very bright, we maybe don't need a big sophisticated microscope. So here we teamed up with engineers from Los Angeles, Aydogan Otcan lab. And so he's transforming smartphones into portable microscopes. So you have a little device, a click-on device. It takes electricity from the smartphone. It uses the screen, the apps, the processor, and the camera of the smartphone. So you have a little laser pointer in here. You can buy for €5. You can buy lenses in the internet, which you put on your smartphone, and they make it a microscope. So you only put between the sample and your smartphone camera is this lens and a filter, and that's it. And so the idea is, if we can make dye molecules so bright, can we make single molecule detection even on a smartphone? Can we make fluorescence assays more sensitive if we use this fluorescence enhancement? And so that is very simple how it looks. They have a laser, neutral density filter, and then one lens which focuses is here on our sample. So it goes a bit, it's not collected. I mean, the NA of the smartphone is of course very small, so it's very easy to have like a dark field illumination just by coming from the side, and then you detect what is here on the surface with this lens and the filter and the smartphone, so it cannot get easier. So this is how we built it on the benchtop. It's not integrated yet in this click-on device, but with these new cameras, it's almost shocking what you get. I was first telling my students that he confused some files and measured it on a microscope, but he was very sure he did not. So does it start like this? So this is a spot. So we didn't put one molecule, because one molecule can also be a piece of dust flying away, whatever. So we put like 3 or 4 molecules. 4 molecules we put into this hotspot of this DNA origami antenna, and this is not published. But I was so almost shocked when I saw this. Where's my pointer? I think it's not starting automatically. So this is detected just with a smartphone camera. So if you look at this guy, bam, then it's still shining. So now we are here, and then it goes 2 more steps. So it's normalized to the first step. Otherwise, you would also see a couple other molecules which go like this. But you can really see all these single molecule steps very easily with a smartphone, and this is almost shocking how that works. So this guy is a super bright guy here. So this is another one that is sitting here. So yes, it's in a few years maybe we can do it all on a smartphone and don't need any sophisticated microscopes anymore. So I think I should stop with that and quit our force clamp story, and instead thank all the people who did the work. So it's a bit reorganized. This transparency is not in such a good shape. These people haven't given me their pictures yet. And this is because we are still rebuilding the lab now in Munich, and many people are coming and going. And actually the corporations are more the current corporations, and I should have put also like these people we developed the photophysics with, like Markus Sauer, Mike Heilemann, Jan Vogelsang, Torben Cordes, and many others. So these are more the current corporations and funding schemes sources I'd like to thank, and you for your attention.
So thank you for this very nice talk. So I have a question. Like in this photostability experiment and this, I mean, this radical anion state. I mean, did you check freezing of the sample? I mean, like I mean, if you go down to the liquid nitrogen temperature, or just like -10, like I mean, can we improve photostability by this? Like.
Well, we didn't try it. So the simple answer is no, we didn't try it because yes, we want to do it all under more or less close to physiological conditions. I mean, there are other groups who have recently done a lot of cryo microscopy also for photostabilization issues and for stability, and they show that it gets more stable under those conditions. So yes, the photostability goes up. So Jörg Enderlein's lab and Sandokter lab, for example, they built this. They tried to make this cryo super resolution microscopy.
Any more questions? Oh, sorry.
Thank you for this very nice talk. I have a question about the mechanism you suspect when you have this loss in proportionality between lifetime and contaminant in the self-quenching. You know, when your dyes are close together.
Yes, yes.
Okay, and the lifetime gets.
Yes, it was puzzling
Yeah.
that the lifetime changes after the intensity. So when you put two dyes closer together, it seems like they were first getting darker, and then the fluorescence lifetime was going down. So what we suspect is we have an equilibrium between two states, and so most of the quenching is static, and this is because we have this. Well, one needed to show that when we have the origami, and we have all these strands next to each other, and then we have the dye on the one side, and we vary this distance to the next dye. Then we have to fill here the base pairs in between. And when you have the base pairs in between, there are like 3 base pairs, and this strand comes from the neighbor. We believe that it's unbinding and binding. And this unbinding and binding is causing a blinking. This I did not show. And this blinking is like switching between a bright state and a state which is almost completely dark. And this is why the lifetime doesn't change, but the intensity changes. And then this gets longer with longer distances, and then this stays there for a longer time. And when it stays for a longer time, you see the lifetime of the state when these dye molecules interact longer. And this has a short lifetime. So when they really interact, they're not completely dark, but they have a short lifetime. And before you have a blinking additionally by DNA binding and unbinding. That is causing that. So it's a mixture of things that are happening. And it's, so we believe that this is what happens, but we have some controls which show that. Another interesting experiment is if you don't have a double-stranded region between 2 dye molecules, we see that even on a distance, if you have 10 base pairs here and it's single-stranded, we see that they can touch. So this origami is very dynamically breathing. So even 10, it's not stretched by the neighboring bases. It's super flexible and it will touch. And it still they still influence each other if you don't have double-stranded DNA.
Okay.
I have a question about DNA break. Maybe I did not get it completely, but when you want to visualize proteins in the cell, how does it work? You label an antibody with this DNA piece, oligonucleotide, and which is coupled to a fluorophore. But then the antibody size is quite big. So what happens with the resolution, especially?
Exactly, that's a problem. I
cell?
mean, this is, especially, I mean, we did some experiments already before Ralph went to the United States and showed with antibodies you could do that, but you lose the resolution of the size of the antibody. So at the end of the day, it would be great to have direct labeling of these self-labeling tags or something which is not so big. So in the moment, that is, of course, a problem. And you would need to develop like a labeling strategy with DNA without antibodies. But you're completely right. And if the DNA is on the antibodies, you lose 10 nanometers right away. Yes.
I want to ask about how to about the bleaching things. So then you put some antioxidant or the reduction reagent. Then so you could see that apparently the bleaching is reduced. But actually, this is if you are going to illuminate in the same intensity, you can see more longer illumination. Longer absorption. Longer emission.
Right.
Sorry. But if we are going to think about the photon budget, so that means how many photons they can emit before the bleaching. Do you think still it's helpful? Because this is the most important part.
For the photostabilization?
Yes.
Yes, absolutely. That's what I'm talking about. Like in our papers, I think we never talk about photobleaching lifetimes. We always talk about photon numbers. That's all that counts, photon numbers. Yes, so the photon numbers go up. That's the whole point. Yes.
Okay. Thanks.
Yeah, thanks, Philip. Could you make a kind of a statement? How applicable is this ROX strategy to fluorescent proteins?
Oh, well, you're the specialist on
No,
this.
no, but I want
That's
to
not
hear
fair
your
asking
opinion.
me. That's not fair.
No.
Well, so that's I don't have a short answer to that. The short answer is in our hands it didn't really work. We found that with thyroids we get better results when we induce this blinking and do localization microscopy. But as you told me and some others, that under certain conditions with certain proteins it might help, and that you also see the triplets and the radical anions, and that you could probably do something similar. But I don't have a good answer. So the first we tried briefly, it didn't work. Then we did some more experiments, and it might work to some degrees for some proteins, but it will depend on accessibilities and all this, and on the real role of GFP, which people suggested that the historical evolutionary role was an electron donation, but you know much more about this.
That's actually not true, but I wonder if this is actually too naive to believe that this could also work with FPs, these strategies, because we never tried at the single molecule level, but you have more experience on that. So maybe what we see at the ensemble level is just
yes, maybe we should
not the real life.
Design some experiments which are more conclusive. So yes, we have one paper only on where we made a nanoruler with fluorescent proteins, and we see an effect, especially of thiols, but we did never see such an effect of only like ascorbic acid or methyl biology, and it was rather bleaching faster. We also tried on the single molecule this redding, and in our hands it didn't work so well, so maybe we were just not smart enough, I don't know.
Actually, I have another quick question about the self-healing dyes. So it's based essentially on Trolox, the ones that have been...
There were all COT, so it's different mechanisms, but the NBA, Trolox, and COT, these are the three substances that were tested most.
And not thiols.
Thiols, I don't know actually.
Because you explained to us that they have this Germination Recombination thing. So it would seem natural that they would be efficient.
That one would try with thiols. Actually, I don't remember seeing something with thiols yet. That's a good point. But also they might then add. I mean, you also see with other dyes, not only with the polymethene, with thiols you see these long offstates. And I'm not sure whether that will be completely reversible at some points when they're so close together. But I'm not sure. One should probably look more closely at that. That's definitely true. So also like ATO647N, if you have a lot of thiols, it will also go to a long off states, and you can switch it on again, like Sci5. So many of these dyes. So Markus Sauers did a lot of work with Mike and showing that. And it's not for each dye completely clear what state it is, I have to say, from my point of view. There might be this double reduced state, there might be additions of thiols as with Sci5, but I don't know all the details of this. I think nobody yet knows, I guess. I don't know.
Thanks.
Okay, any more questions? If not, thank you very much once again.